So I haven't posted your first assignment in Wiley Plus yet, and when I do, it will show up in the module 1 as a link. It'll say Wiley Microbiology because that's the topic we're on right now. I've kind of delayed in posting that because when I post things, you guys click on them and you do them. But I want to guide you through how you should do these assignments, and we haven't finished the topic yet, so I don't expect you to do it yet. So once we finish the topic, I think I'm going to deploy it for you guys. But I was playing around, right? This is a relatively new program for me, and I was trying to figure out the settings and stuff like that, and kind of by accident on Friday, I figured out I can see it as a student. Other programs sometimes that they give us, we can't like engage like a student does, truly how a student does, but apparently I can. So I played around with it this weekend, and I may even do a separate video of how best to use Wiley Plus for you guys, and then post it for you, and then also in class ask any questions you may have. So how are we doing at this point as far as reading the book? Have we gotten there? Okay, good. I see more yeses than nos than last time.
Like paper and stuff like that, that's on Wiley Plus.
Mm-hmm.
We're not like going to write a paper or anything. That's just like part of the book, right?
Oh, yeah.
I was like, what is this?
Yeah. It's it. The ebook's got all kinds of cool stuff. And remember, no, I haven't gave that lecture yet.
I was looking for assignments and nothing.
Yeah,
like an
they're
assignment.
not. There's no assignments. I mean, you can kind of practice some stuff, right? So actually, I'm kind of glad you asked that, and I'm going to just go ahead and hit this link. And remember, it gives you a thing to open in my web browser. I don't have it open as a whole separate window, but another tab. You can set that in your browser. So, here's the book, right? So there are things like down here she's talking about, like mini paper activity, right? Focus on research, practice quizzes, right? They are just that, practice. Flashcards, right? If you find those helpful. I don't assign those. I could, but I don't just yet. I don't know if I ever will. Like the podcast that the book relates to, and, some of us like that kind of cool stuff. I think that one's kind of interesting. And actually, so this is a quick way, too, that you can get to the animations. And one of them that I do suggest watching, because it's related to today's lecture, is the classification system one. It's kind of a good one if you've watched that. And quite honestly, I haven't watched the other 3 yet. So I don't know if they're good or not. I didn't have my laptop with me last night, and I was out on the soccer field. So I had my book. So guess what I was doing? What you should be doing. I was reading again for the 5th time at this point, right? So I don't tell you to do anything I wouldn't be willing to do myself, right? And I teach 5 classes. So those of you guys that have 5 classes, if I can keep up with it, I expect that you can too, right? If I can't, then I know I'm giving you too much work, right? Okay, so microbiology. So we got through the diseases last time, hence why we were able to do our little poll. So we're starting here on slide 9 with our next objective. Can someone who's near the door close it for me? I forgot to do that after attendance. It might get noisy, and I also don't want to be noisy for the... I don't know if you guys noticed that computer lab. The glass doesn't go all the way to the top. So noise carries to that room. That weird room. It weirds me out. I don't know, with all that glass. Which sucks because people during exam time don't realize that. I mean, why can't they just put some glass at the top? They'll walk down the hallway yelling on their phones. And it's not like they mean to be rude. They just think that they're not being rude, I guess. I don't know. But I try not to. I also found out the other day when I sat in there because I didn't feel like walking all the way back to my office to get to a computer or pulling out my laptop. You do need the new photo ID to use that lab. So if you haven't gone and got your new student ID, which is in the annex building behind building 1, behind campus police, if you know where that's at, that's where you get your new ID. The lines have died down now too, so that's the other good thing. So if you want to go and get that done. My public service announcement for today. And speaking of which, did I tell you guys about single stop? Did I ask you guys about that? I did tell you about that too. So our boss, even in our meeting, which you want to talk about following directions. So we have a meeting once a week, departmental meeting, and sometimes workshops and talks and all that good stuff on Monday afternoons. And we always have a sign-in sheet, right? It's usually just a piece of paper, you sign your name. This time it was printed out with all our names on it. And somebody signed it, and the next person signed it, and the next person signed it, and guess what it said at the top of the page? It said office location and phone number. He wanted that information from us. Yeah, we passed the paper. And when the paper got to him, he was like, you complain about your students not following directions. Who in you complains about that? Because y'all just did it. You know, so we all get distracted, right? And but one of the things is that, you know, we get bombarded with information, don't we? And communication is one of the most difficult things at Delgado. It just is. I think when you're big and you just don't know how to handle that, it makes it harder. And then with the 8,000 different ways you can communicate with people nowadays, they're really begging us to help more with communication because we've got your attention right now, don't I? Gotcha. And our Canvas sites are a really great place. Like I said, they're already blasting you on the front page, but students skip the front page too. And I don't want to like bombard my Canvas site though with information. What I'm going to probably do is a wiki page, and the title is going to be "You Need Help," and I'm going to list a whole bunch of resources there. But I am going to try and do like one public service announcement until I run out of them, and not do too many each class. Okay. So if you run into something on campus that you think is important that your classmates should know about, and you want it to be the public service announcement, I'll even let you do it, or you can tell me and I will tell the class. Right? So that's how we work, right? We help each other out in this very complicated world we live in now. So the next thing we're going to do that can be complicated is talking about science and defining science and sciences and what we do in science. And unfortunately, at some point, somewhere in our lives, unfortunately, some teacher told you that you absolutely have to have the right answer. And unfortunately, I'm going to give you tests where there's pretty much only one right answer, apparently, at that time. See the problem with that? That's actually not how science works. Science is an investigative method, what we refer to as the scientific method, right? And we observe, we gather evidence, we gather data, and we make assumptions or hypotheses, or we make theories about what it is that we've learned, right? And we make conclusions based on that. And we do this, you use a scientific method every day of your life, right? In decision making, for the most part. It's actually a natural human process for most people. But like I said, then we get this point where this is taught out of us, right? We need to find it again for some of us, right? Because that's not the best way to learn science. And in fact, there are other studies out there that you think that like, okay, right now this is the correct answer and it's never going to change. Not true. My mind was blown when I was in grad school. My ex-husband was a history major. History changes. You're looking at me like, what is she talking about? History changes? It does, right? Because we base it on data, information that I don't know about you, but I wasn't alive when FDR was on this planet, right? That president, right? I just like Roosevelt. Franklin D. Roosevelt. Blanked on his name. Again, we use acronyms a lot, don't we? And then we forget what they stand for, like DNA. Kind of need to know what it stands for, though. It's important. So we need to get back to that, right? And so, yeah, we know about stuff, right, because of what we've read or what was documented and things like that. But as more stuff, right, some weirdo in his attic had letters from somebody, right? And we discover them and we're like, oh, wow, that totally changes what we believed about what happened back then, right? So the same thing happens in science all the time. Like when I was sitting in your guys' seats, which was unfortunately a long time ago, some of the stuff I'm teaching you now, didn't even know when I was in your seats, right? We didn't have the ability we didn't have the tools available, we didn't have the resources available, we didn't have the brilliant minds at that time to discover the things that you guys are learning about. Right? So it's constantly changing, it's constantly evolving, right? Sometimes a dirty word. It's not a dirty word, it's a scientific word. Evolve, right? There is evidence that evolution has happened, right? To what degree or extent, right, can be argued. And that's what should happen in science. And true scientists, like when some other scientists publish it and they don't believe in it, they die their damnedest to disprove that other person. pretty much like in everything else in life, right? So our definition of microbiology itself even continues to evolve, right? So do I expect you to memorize some specific definition and regurgitate it back to me? No, for one, that means you probably only memorized it and you're going to just throw it out when you get done with this class and have learned nothing from it, right? Instead, we need working definitions, definitions that you understand, right, that help you with the verbiage that we use in science, right? And so some of us already can pick apart even the word itself, right? Micro stands for, or is, small. Biology is what? Study of life, because ology means study of, bio means life. So we're studying small life. That's in the simplest form of the definition of microbiology. But in addition, right, we use certain methods and stuff to be able to study microorganisms that have been encompassed under the definition of microbiology, right? So other ologists, right, other people studying other fields in science and even in other fields like anthropology are doing things like DNA testing, right? Most of the time you think, okay, DNA testing is science. Anthropology is maybe science, maybe not. It's an ology, study of, right? So these things are becoming intertwined in our lives, right? But that's a microbiological, actually molecular microbiological technique, being able to analyze DNA, right, and infer information from it. So it's very complex. It's getting even more complex, right, as we go further and further. So a definition probably even different from the one that's in your book. Just, again, to get us working with a general understanding of these terms that we're using. So typically it's defined as a study of organisms too small to be seen clearly by the unaided eye. In other words, you can't see them without the use of what? Microscope, right? The microscope is the most common tool used by microbiologists. Techniques that we employ, growing bacteria, especially, DNA stuff and stuff like that fall under those techniques. Petri dishes, right, which were designed for working with microorganisms, right, are a microbiologist tool now that's used by multitudes of different types of people for different reasons. Are viruses alive?
No.
Most scientists would say, just as you did, no, right? But do microbiologists study viruses? Yes, they do. They do. They do. I don't. Although, I guess if I could do it all over again, I probably would, because they're pretty cool, right? But there's another term you'll see used in your book when we want to talk about microorganisms and viruses and not say that viruses are microorganisms, that they're living. Anyone know what that term is? So we're not going to call it a microorganism. We're going to call it a microbe, right? When they say microbe, that's the most general term for something microscopic, right? Could be a bacteria, it could be an algae that's microscopic, a protozoa, it could be a virus in that case. So that's a term that's really getting hot right now, right? Because we want to just be able to say one word and not say microorganisms and viruses. And you can even go into subfields of microbiology, right? So if you just study viruses, we say you're a what? You're a virologist, right? You study virology. Or if you just study bacteria, you're a bacteriologist, right? If you just study fungi, you're a mycologist. Which most of my A&P students go, what did she just say? That sounded like muscle to me. No, myco actually means fungi, right? And so you're studying fungi, right? Molds, yeast, mushrooms, those kinds of things, right? And there's like a gazillion other subdisciplines, right? Those are like just the major ones. So this is all encompassing. And in general, right, microorganisms are going to be teeny tiny, and they're going to be relatively simple, because they're so tiny, and they tend to lack. Now, this is the other caution when it comes to science and in life in general. Do most things fall into the classification of all or only? No, there's always exceptions, right? And so if we say all or only in science, rarely is it true, and rarely is it true for very long, right? Always be wary of all and only statements in science, right? 9 times out of 10, it's false. So a perfect example of an exception to this one is the rotifer, right? If you search on the internet, you can find pictures and videos. You can see, like, microscopic. You can only see him under the microscope. You can get pond water, and you might see this guy sometimes. He's so distinct. He has his own phylum of classification, which we'll talk about in a moment, or review a little bit of that. You can see his intestine under the microscope. So that's distinct tissues, right? It has an intestine. It's microscopic. It's crazy. But, you know, he's the exception. He's not the norm when it comes to that. So this is one of my favorite websites, and this link wasn't working for me yesterday. A student turned me on to this years ago. So I just do a quick web search, right? The internet is a wonderful place if you know where to look. So I want cell size and scale, and it's Utah, and it's actually their genetics division that put together this. So I should hopefully be able to find it. There it is. And so now it's interactive instead of just a screenshot. So they did this really cool interactive. The only thing is their latest version of this is kind of graying out what I really want to show you guys, and that's measurement below millimeters. And so most of us have heard of millimeters. You can see millimeters on a centimeter ruler, right? But in, of course, the microbiology world, in the microscope world, we go smaller than that. And in fact, a millimeter can be broken up into 1,000 little tiny micrometers. So 1/1,000,000th of a meter. And then we go even smaller into nanometers, which are 1/1,000,000th of a meter, or again, now we've just stepped down 1,000-fold. And then things like angstroms and picometers, microbiology, not so much, but with the high-powered atomic force microscopes now, we can go down to atomic level of molecules, right? So they're measuring stuff that small, that crazy small. So let's take a ride. Down here, you could move the little cursor and we can zoom in. So at this point, these are things you could pretty much see with the naked eye, right, if you have normal vision. And then we can zoom in, right? And you would probably need a microscope to see a sesame seed in that much detail, right? And you definitely would need a microscope to see a grain of salt, which is only about 0.5 millimeters, right? And then to see the microscopic organisms like amoeba and paramecium and the human egg, which is the largest cell in a woman's body, or a single skin cell, or the rod photoreceptor in your eyes, right? We would need microscopes to see these things. Red blood cells, to see them in this detail, you'd need a microscope, because they're 8 micrometers in size. You can see baker's yeast, like we did today under the microscope in lab. You could see chromosomes when they're condensed, like the X chromosome. You could see bacteria, which you can see in this instance as compared to mitochondria. This poor little E. coli is smaller than a mitochondria even, right? And he's a single-celled living organism. Lysosomes that you'll find sometimes inside of our cells. So these are parts of cells, right? And tiny bacteria. And then with a light microscope, you're really not going to see these guys. You need things like those electron microscopes, right? Those really fancy ones that we hope to maybe get someday here at Delgado. We've got some really crazy biotech guys that are really good at writing grants and getting equipment, and so they're working on it. It might happen. So viruses, right, actually seeing down to this level wasn't until the advent of electron microscopy and then the more advanced now. And we can go down even further and actually see large structures within cells like ribosomes, which are made of protein and ribosomal RNA and make proteins. Transfer RNAs, very large proteins like hemoglobin, beautiful proteins that I love like antibodies, or the phospholipids that make up the bilayer of your cells. We go down to a single glucose, a single sugar molecule. See its individual atoms with these atomic force microscopes. A single nucleotide that makes up DNA. A single amino acid that makes up proteins. Water molecules, which is just three elements, right? An oxygen and 2 hydrogens. That's it. And then at the core of it all, and there's a reason why they put this baby here, is carbon. All those things, right? At the center is carbon. Carbon is the backbone for sugars like glucose, amino acids like that methionine, the phospholipids, the nucleotides. They all start out with carbon atoms and then other atoms like hydrogen, oxygen, and nitrogen, and phosphorus, and things like that attached to it. But it all starts out with carbon, and this is why we can ask the question, with carbon dating, how long ago was it alive? Because we know the decay rate of that element. And that's what they're doing when they're carbon dating, right? Because when it's alive, that element stays maintained, right, when it's in a living organism. And then when you die, it starts to decay at a particular rate. And so depending on the decay that is in that carbon, they can determine about how long ago it was alive. Because we are carbon-based life forms on this planet, we haven't found anything else as of yet on this planet or any other planet in this solar system or the next. Could it possibly be something else somewhere else? Or even on Earth? Someday, maybe so. But as it stands right now, it's all carbon-based. It's all about carbon. Kind of cool, huh? Go back to presentation mode, Erica. So the next thing that scientists do a lot, not to say that others don't, is we group and classify things based on similarities and differences. So one of the major defining things, and if you watch that animation classification, when they were looking at things under the microscope, they saw these distinctive differences between cells. And so they divided them up into 2 groups based on this difference, prokaryotes versus eukaryotes. And then as we continue to learn about more and more organisms, we started grouping them in even more groups. And at one point, 3 very important groups were established for living organisms. So this doesn't include who that's not living, that's a microbe? Viruses, right? So this is for living organisms. We have 3 domains. We have archaea, bacteria, sometimes known as true bacteria, and eukarya. You'll notice that 2 of these words seem similar, right? Eukarya and eukaryotes. And those of you guys that are really super spellers, and congratulations to you, start to get annoyed when they see some of my slides because they think I have a spelling error. But believe it or not, this time I don't. And I left it there on purpose. Anyone going, Miss Erica, you spelled prokaryote and eukaryote wrong? Nobody? Yeah, isn't it supposed to have a K? Nope. It can be C or K, just like my name, Erica, can be either a C or a K, or in the case of this Erica, the first time I've ever seen it, parents couldn't decide, put both of them in there. Both C and K. Covered both bases. Both are correct spellings of this word, believe it or not. So the C or the K is accepted. For right here, right? For karyote. Anyone know what karyote means? Who read their book? Prokaryotes are single cells. Yep. Prokaryotic cells tend to be single-celled organisms. That's true. But what does karyote mean? So, well, first we've got 2 things have karyote in it, right? One of them says pro, which means what? Before. The other one is eu. Anyone know what eu means? True. True. So one had a true karyote, and one they believe became before the karyote.
Define nucleus.
Yeah, the nucleus looked like a kernel to them under the microscope, right? It's in the center, it looked like a kernel. So that's actually what karyote means, is kernel. And so they believed eukaryotes, which had it, right? So they had a true nucleus, as opposed to prokaryotes they believe came before and did not have a nucleus. So that's the defining characteristic between these two cell types. One has a membrane encased, defined, delineated, whatever adjective you want to use, membrane, right, that defines what we call the nucleus. The nucleus is an organelle. It's an organization within a cell. Bacteria are prokaryotic. They don't have that. Prokaryotic cells don't have, you're not going to see a nucleus under the microscope. Even with the electron microscope or scanning atomic force microscope, no nucleus. Okay. What's typically in the nucleus of a eukaryotic cell? What type of molecules? DNA. Okay. Prokaryotes, do they have DNA? They do have DNA. Bacteria, prokaryotes, archaea, these have DNA. All living cells have DNA as their storage of genetic information. All of them. So far. No exceptions just yet. What does this mean then? It's just not encased in a membrane. That's all. They just didn't put it in a membrane. And in fact, eukaryotes put it in two membranes. It's like serious. So eukaryotes tend to be more complex. So not only do they have that organelle of a nucleus, they'll tend to have other organelles. So can you guys think of any organelles you learned about in biology class that we may even saw one of them on the screen earlier? Mitochondria. What about a plant? Chloroplast. What does ER stand for? Endoplasmic reticulum, right? Which can also have ribosomes attached to it, so then we call it a rough ER. Because it looks rough. It's got little studs on it. It's so cool. Okay, so what else? Any other ones? Whole bunch of stacked membranes, packages, stuff. Golgi apparatus, lysosomes, right? All of those are membrane compartments. All of them. Right? Only found in eukaryotes. So at one time, and I don't know how much we've deviated from this, there was a system of organizing organisms. And of course, plants and animals was easy, right? Animals move around, they eat stuff. Plants stay still, they photosynthesize. Then you had these organisms, and literally they got this name because they did not fit in either one of those categories, right? So you like Euglena, which we didn't look like in lab this week because I don't have any good slides. He photosynthesizes and eats and moves. Crap. He's not, is he an animal? Well, he's moving and he's eating, but he also photosynthesizes. So is he an animal or a plant? He's in between. He's protesting. He's Protista. It's actually where the name comes from, right? They protested. They didn't want to be put in either one of those two groups because they don't fit. They have their own group. And then fungi, most of us are aware of that word, right? So where did bacteria fall in this world? Monera. Monera is where most of the bacteria were. So you learn in school to write this classification scheme where kingdoms are at the top, right? These big overarching groups that we just talked about. And then you learn about a classification below that. Anyone know what the name of that grouping is called? Phylum, right? And then we keep going, right? And so when I was in school, there was a little saying I memorized, a mnemonic to help me, which was, Kevin, please close our front gate slowly. So it gave you kingdom, phylum, and I always have to go through it. Kevin, please, right? Close, class, hour, order, front, family, gate, genus, slowly, species. but I'm going to say a curse word for some people. I'm going to say a bad word right now. We actually have a classification now above that known as domains. So now I say, damn, Kevin, close our front gate slowly, please. Front gate slowly. Right? Damn. D for domain. Do you have to memorize that for this class? No. But you do need to recognize domain as above kingdom. And the three domains are archaea, bacteria, and eukarya. And you'll notice that they're somewhat linked to cell type. Who's ever heard of archaea before today's class? Not many of you, right? So at one time, these guys were lumped in with bacteria because we thought they were all the same, right? We didn't know that much about them. And then come to find out, they're actually quite different. And they actually might be more of an ancestor to eukaryotes than bacteria are. So he's kind of like in the evolutionary tree of life, right, that we're trying to figure out because no one has a time machine, right, that may have been a split from bacteria and archaea, and then we split off of archaea, they believe. So when you look at the tree, it should be my next one. Yep, there it is. Stolen from someplace on the internet. Borrowed, shared, educational purposes. I'm not making money, right? I'm trying to figure out what to do about copyright stuff right now. I probably got to go talk to my librarian friends. My boss said, you put your stuff on YouTube? I'm like, yeah, that's what they told me in the workshop here at Delgado. Because you guys don't have enough storage space. And when you do that, right, it can be out there in the public domain, even though I thought I had hidden it, but apparently it's not hidden, all that jazz. I don't make money off my YouTube channel, right? If you make money, right, then you can get in trouble with copyright when you use other people's stuff and claim it as your own. I don't claim this as my own, right? Not these parts, not when I use someone else's pictures, right? And I'm using it for educational purposes. So I fall into that little gap of I shouldn't get in trouble. Shouldn't. Try not to get in trouble. So you see what I mean, right? There's this common answer so here, and so here's the split, bacteria, archaea, and then somewhere down the line came eukaryotes, which of course we're over here, right? Which kingdom do we fall under that's listed here? We're animals, right? We like to put ourselves on this little pedestal, right? We're animals. We really are, right? And then of course fungi and plantae are over here. And then weird names you probably never heard of before, right? Over on these other branches. And some we'll talk about, some we won't. So who came up with this? Carl Woese is one of the guys you need to know what his contribution was, and this was his. So back in the 70s when we started the laborious process of being able to look at sequences of nucleic acids, he started looking at RNA sequences, specifically the RNA associated with the ribosome, because all cells have ribosomes as well, because they need them to make proteins. And so when he looked at this, that's how he came up with these three major groupings, right? Everything started grouping together in these three major groups. And so again, when I was sitting in your seats, wasn't in the textbooks yet. Hadn't gotten to that level of acceptance in the wide scientific community of the world we live in. But it is there now. Carl, go Carl, although he's probably dead. All right, so what do we need to know about these groups? Well, we need to know some characteristics so we can understand also, other than looking at the sequence like he did, what are some similarities and general similarities and differences that are amongst these groups? And so for me, when I got to do information like this, I'm a table girl, right? I put a table together. And sometimes I forget which way I want to go in the table, so you notice the little blocks that are going to disappear in a minute are numbered. That's my trick. Okay, so we're going to start with cell type. And so what are the two choices for cell type? Prokaryotes and eukaryotes, right? So anyone know what bacteria are? Who's been paying attention? Because I've said it. Prokaryotes. And archaea? Prokaryotes. And eukarya. Oh, they're named the same. That's nice. Cell organization. I've said this a little bit, right? Unicellular, which means a single cell is the organism, or organisms can be multicellular. For the cell type prokaryotes, they're only unicellular for the most part. We have some that we're like, ooh, Are they blurring the line here, right? But for the most part, they're unicellular. What about archaea? Right, all prokaryotes at this point, like I said, are generally unicellular. But what about eukarya? We already said animals and plants fall in this category, and they're definitely what? Multicellular. But are there some unicellular eukaryotes? Yeah. Amoeba, right? Euglena, right? These guys fall in that category. So, or. Could be either one or the other. So a defining characteristic, although we know plants have cell walls, their cell walls, anyone know what substance they're made out of? Cellulose, which is actually glucose hooked together in a particular way that's really strong. So strong you cannot break that, and I talked about that last time, right? That's why a kernel of corn goes in one and out the other if you don't chew it. Break that cell wall. There's one group here, their cell wall, and most of them have a cell wall, is made out of peptidoglycan. Anyone know who has that particular substance? Bacteria. Bacteria. So yes for this one, no for all others. Not to say they don't have cell walls, right? We're prefacing it. Who's got the peptidoglycan? And we're going to talk about that substance a lot. You'll hate it. No, you won't. I'm going to make you love everything. Okay. So membrane-bound organelles. So the nucleus is a membrane-bound organelle, right? And then we named a couple others just a moment ago. What cell type has those? So which domain? Eukarya. Do prokaryotes in the domains bacteria or archaea have membrane-bound organelles? No. Where on Earth, what environments would you find bacteria? Everywhere. You name it, all of them. You probably find a bacteria. If you were searching for archaea, where are you predominantly going to find them? Very hot, very cold, very salty. So we call these types of environments extreme environments. What about, in contrast, eukarya? Moderate. Basically anything that's not extreme, right? Like me. I'm Goldilocks, you know. Porridge has got to be just so. Not too hot, not too cold. Not too salty. Why is my... Okay, I don't know. So remember, this is microbiology class, right? Although we're going to talk a little bit about animals because we're going to talk about ourselves, right? Because we can't stop that. Right, it's human nature. We're going to focus more on the microscopic, right? We'll make some comparisons sometimes to plants, but we'll talk about those protists like I talked about, those unicellular algae, protozoa a little bit, we've already mentioned. Slime molds and water molds, not really my forte, but interesting little things. And fungi, right? Most of us are aware of fungi and we like the cool things that fungi can do. Most are larger than prokaryotes. Again, notice the term most, not all, right? There are some exceptions. There are some eukaryotes that are teeny tiny microscopically, and there are some prokaryotes that are pretty big microscopically. But for the most part, these are the bigger organisms. As we found out in lab this week, right, we could see them pretty easily next week when we start doing bacteria. Tiny. Oil immersion from now on. So viruses, again, until we got things like electron microscopes and stuff like that, we really start to see their structures and get a better feeling for them. And they're said to be acellular. Of course, when you put A in front of the word, it means not, right? So this is not a cell. Viruses are not organized to the cellular level. So that's my number one reason why I don't consider them a living organism. They're not even a cell. But they contain things that cells have, right? And we'll talk about that in more detail when we get to our viruses topic in Module 2. So they can be, they're microbes, right? That's our encompassing term now. They can be 10,000 times smaller than the typical bacteria. And again, I think that website that I went to where you can, you know, interact and zoom in, you can see how tiny they are in comparison. Why are some people obsessed with them? Because as we know, many of them cause diseases. Some we talked about at the beginning of this course. Others can cause cancer. And what cancer is, in simplest terms, I find to be able to describe to people, even non-sciences, is your cells behaving badly or not doing what they're supposed to do, right? That's it in the nutshell. And so since viruses have to get inside of cells to use the cells, ribosomes, and machinery to copy themselves, they can disrupt cellular processes in that process and cause a cell to go awry. So the problem with cancer, and my husband is one of those non-sciences people, I love him to death, right? His mom died of cancer, so he has an emotional component attached to that. There are not mean people, I don't believe, in this world holding back cures to certain cancers. I really don't believe that. Again, cancer is your cells behaving badly or not doing what they're supposed to do. How many different types of cells do you have in your body? A lot. I don't even know the number. I don't even know if it's hundreds or thousands, but it's a lot. Definitely a lot, right? Probably in the thousands, maybe, right? So each one of those cells has a job, right? Has things it's supposed to do or things it's not supposed to do. And so when the cell gets disrupted, either by a virus or something else or something genetic or environmental or any number of things or combination thereof, that's when cancer happens, right? So just you somewhat understand how this one word is encompassing so much stuff, right? Like too much, really. Too much. And for someone that's not educated, right, you can very easily be misled.
Aren't most viruses, whatever, diseases, anything, aren't most of the time like dormant until like
You're talking about latency maybe to a certain extent, and we're going to get to that when we're going to do a whole topic on viruses in Module 2. So I'm going to say hold that one for now.
Do we need to know the sizes of the bacteria?
No. Just relative understanding of how small they are. Okay. So then we get to this handsome devil, Anthony. I love the wigs they wore back then, huh? And so, and again, I put this date here just to try and kind of help us gauge, right? This is not a history class. I hate history classes that make you memorize dates anyways. Just want to let you know this is, like, I don't know about you guys, I have trouble fathoming how long ago this was, right? This is like 300 years ago now. Yeah, I'm just like definitely not anywhere near our I'm not a vampire. I've only been around for like 40-something years, you know. We don't have time machines. We don't get to go hang out with this guy. Why do we know, and why do we give him credit, though, for being the first person to observe? And he may not have been the first person, but he did the next step. He documented, he described, right? So this drawing, which I found out recently, too, he didn't draw. He hired someone to do it. But he wrote letters to the Royal Society in London, right? He was probably the first to accurately describe Giardia. Why? Because he was looking at his own poop. This dude didn't just stop at looking at his fabric because he was a drapery merchant. He looked at everything and anything he could get his hands on using the lenses that he made, right? And he made some pretty amazing lenses. The one thing we really wish he would have wrote down is how he made those lenses, right? He did not share that, right? That was his secret that he didn't share, you know, that we all wished he would have shared. I saw it and heard it. Take it back. So, look at this drawing. In a second, once I make that go away. I didn't make it in time? Are you really that awful? Or did I bump something?
Probably just unplugged it.
Crap, because it's not on, I'm not recording, though. That's the one bad thing. So shut up, Erica. What time is it? Figures. Okay. All right. Are you still going? No, you're ticking, so you're good. Okay, so look at this again. Now that we can see it. Okay. Little circle, little circles, not semicolon. Figure E is little circles, right? Notice the others don't have semicolons. Well, I mean, they do, but they're tiny. See, over there, not next to the letter. So and this is the one that moved. We went over there. Okay? So again, we know this because he wrote letters. He hired someone to draw this for him. We have them in some museum somewhere.
What's going on with that? Just curious.
Hmm?
Like weird
Oh, F.
lines and stuff.
F. Oh, lots of different sizes of
Oh,
rods.
okay,
Yeah.
that was
We'll talk
fun.
about, yeah, stuff like that. And this one's kind of wiggly, you see? Probably not a worm, by the way. I mean, like, he was probably seeing bacteria, y'all. You know, I mean, he was really amazing lens maker. I didn't show that. Alright, so this is actually the last thing we're going to start but not finish before next week. So, other thing you'll notice that I probably to a certain extent need to change some of my objectives. Can you discuss or describe on multiple choice true and false matching test? No, you're going to distinguish and you're going to identify, right? But really in class we're going to discuss and describe. And so that's a higher order ability, right? So can we step down on the test? Sure. But if I just identified or distinguished for you, could I then ask you to discuss on a test? That wouldn't be really nice because I didn't let you practice that higher order skill, did I? Right? And again, we don't do those on my test because I don't want to grade them. Although I did it one time because you guys could type on the computer and I could actually read them. It stressed my students out, right? And I understand that. I have that same level of stress and I want you guys to perform well. But we want you to be able to write too. So where do I have you write under lower stress conditions? Discussion board. So remember that when you're writing, please try to not write like you talk. Does that make sense? Like actually try to write. Yeah, or you text talk. Yeah, please. And I forgot to say that before, and you guys have already started working on that discussion board. But again, in the beginning, I like to warm you guys up to these things, right? And give you some time to practice. So there was this historical concept of what's called spontaneous generation. So let's pick apart the word again. When somebody says spontaneous, what does that mean to you? Spur of the moment, right? Just poof, right? Just going to do it. Generation, history of your family, where you come from, right? Growth. Words like that come to mind when someone says generation, when you're trying to define generation. So there was this belief that this bottle could turn into a baby. You're all laughing. I guess that's good, because that can't happen. I'm not David Copperfield or Chris Angel. It ain't happening, right? And that's not real either, right? That's illusion, or what they call magic. Okay. Can a steak turn into a maggot? Nope. Nope. Steak does not turn into maggots. Eggs from flies turn into maggots, but those eggs from the flies, do you always see them? No. So people thought steak, maggots, right? Steak turned into maggots. Not the case, right? So unfortunately, some of these scientists are left out in your book that help disprove this belief, right, that things just went poof. So again, I got them listed, all the guys that we're going to talk about. Yeah, there are some girls out there, by the way. It just took a while before we got on the scene. And then some of us didn't even get credit until years later, after she dies. Just to give you an idea, again, this was a long time ago they were having this debate, right? So that thankfully nowadays most of us laugh when I say this can turn into a baby. Right? We know that not to be true. Right? So, living organisms can develop from non-living or decaying matter, right? Things could just poof. So I think Louis' experiment is the only one that's in y'all's book. But how did he get to where he was? There were people before him, right? And sometimes that's important. I think definitely in this discussion on how he got to why he did the things he did, you got to talk about what the people before him did that he knew about, right? And why he designed his experiment as he did to prove to all those people who didn't believe, people like himself, hey, right? It just doesn't go poof. It comes from somewhere. And it was pretty easy with the whole steak and eggs, haha, right, maggots, thing. That's the larval form, by the way, of a fly, is a maggot. It goes egg, larval, fly. Adult fly. So I've got some pictures. We're going to go over it. Even though these guys aren't in your book, right, you're responsible for them for the test. I added those questions to Wiley Plus, right, when we get to that. And, yeah, I'm done for today. How about you?
So we have to know.
Yeah, we're gonna go over them. That's our next lecture. Mm-hmm. Yep. Be able to match them up with their experiment, a description of their experiment. And we're going to talk about Koch's postulates too, which remember, they go into more detail in chapter 18, right? That section you should read.